So this is the Unit 3 Summer 2018 exam, and they've got an experiment where it says you've been asked to find the optimum concentration of the enzyme pectinase. So it's an enzyme experiment. And what's the enzyme? It's pectinase. You can tell it's an enzyme because it has A's at the end. And what does this do? Well, if we've got a drawer over here, let's draw some apple cells. There's a cell wall. And there's another cell wall. And let's just draw one more. So apple cells, plant cells, and they have cell walls. These cell walls are essentially glued together by a substance called pectin. So that is our pectin, and that holds the plant cells together. If we're going to get some juice out of these apples, so the juice is inside, then we need to break apart these cells and separate them, and mash them up and get the juice out. So that means we need to get rid of this pectin, and that's what this pectinase done. It breaks it down. So our experiment is, we're getting some beakers and some measuring cylinders. So it says 5, but I'm just going to draw one. So we're going to get a beaker and a measuring cylinder. Rubbish measuring cylinder. And we're going to put apple puree into the beaker. So we're going to have 50 grams of apple puree. So it's been mashed up, but some of these cells are still attached together. So we weighed that out, and in here, this is what we got our pectinase. Let's do that in red. So we've got our pectinase in there. Pectinase. And then what are you doing? You're going to put that into your apple, and that will presumably start to break that down. And we start off with 0. This could be 0%, so no pectinase. Then we've got 0.25, 0.5, 1 and 2% pectinase. So different concentrations of our enzyme. And then what you're going to then do is you're going to stir that beaker with your apple and your pectinase, and then we're just going to cover that with some cling film. That'll hopefully stop any evaporation of liquid. And then after 15 minutes, you get a conical flask, and you're asked to weigh it. So we know the mass of this conical flask, and then you're just going to set up a funnel with some filter paper in there, and then you're going to put the apple puree. So put that apple into there, and apple juice is going to come out down there. And then the apple juice is only going to come out down there if we've managed to break apart these cells with the pectinase. So more apple juice there, the better pectinase has done its job. And then you're just doing it with the different the different concentrations of the enzyme. You might have to do some repeats, think about repeats, and you can record the mass of apple juice obtained.